also the video, a little bit motivating video about the journey of cell. It's a very good video. It's just the simulation of the inner space in the universe in every cell. So you may imagine that cell is pretty small, but everything what is needed for some life is present inside. And today and also next time we are going to talk about cell structure and cell organelles functions. About the structure of today's lecture. So also you received the feedback about the mini test from last time. I'm happy that majority of you received at least some points, which is I think a good benefit to receive the credits. And also now I'm going to start to talk about the cell structure, cell organelles, at the beginning about the difference between okay, where is the presentation, between eukaryotic and prokaryotic cell. Please, could you see the presentation?
Yes.
Okay, great. And after this, after my talk, I will also ask you if you would like to do your optional homework. The optional homework today will be to prepare a 3-minute presentation based which topic you select, and maybe I will show you the topics. That one, okay. Here. So you may think about it during the presentation, during the talk. So the topics, there are few options just to dedicate your presentation to Golgi, lysosome, mitochondria, nucleus, smooth endoplasmatic reticulum, loose endoplasmatic reticulum, or ribosomes. So during my speech, just to think about it, which type is closer to you. And then after the speech, you can decide it, and after your decision, I will also show you one video which is, I think, pretty illustrative and also helping to prepare your own presentation. So let's start with journey of cell structure. And maybe I have to check that I'm recording. Sometimes it happened that I forgot, and it's not pretty good. Yeah, I'm recording. Nice. So everyone has an access to the video. I just put it to YouTube, and also I put the link to Facebook. I send it to the chat of Teams, and the third place which I'm going to put it, it's also Moodle. But Moodle it's now not working, not yet, because the subject which was taught last year as a biology, it has different number, and now I have to renumber it, and I cannot do it personally. I have to ask the supervisor of Moodle. So it takes time, so be patient, and maybe next time we will have also Moodle working. So cell structure. Actually, we have two types of cells in our Earth. We have eukaryotic cells and prokaryotic cells. The prokaryotic cells, they are much simple, and we expect that they were developed as a first one. They contain just the DNA or RNA, which can be called as a nucleoide. There is no membrane which can really distinguish this nucleoid or genetic material from the rest of the cell. The DNA or evenly RNA, it's usually circular, mostly DNA. It's circular, so it has no end and no beginning. Also, there are some ribosomes, and also very important, the cell membrane, which protects the prokaryotic cells together with cell wall. Sometimes we can observe also some capsule, which again increase the mechanical stability of prokaryotic cells. Sometimes we can see that there is some flagellum or some trichomes, or so some organelles which are helping to move. to prokaryotes. Usually to the group of prokaryotes belong organisms which is called single cell organisms. On the other hand, the little bit advanced model of cells, it's eukaryotic cell. Eukaryotic cells always contain or the genetic material DNA in eukaryotic cells is linear. It has a beginning, it has an end, and also the DNA is encapsulated or protected or shielded in nucleus. It's a little structure inside nucleus, and therefore we have this two-wall protection against damage of DNA. Also, the replication and duplication is provided by a little bit different way than the prokaryote. We also have in eukaryotic cell ribosomes. This is the same, which is present also in prokaryotic cell. But also there are some another organelles which are typical only for eukaryotic cells, like mitochondria, GOLGI, endoplasmic reticulum, and so on. First of all, I have to mention that eukaryotic cells are typical for the more than one cell organism. So we have eukaryotic cells. We may distinguish also two types of eukaryotic cells or two models of eukaryotic cell as the plant eukaryotic cell and also animal eukaryotic cell. There is a little difference. The first one is that in eukaryotic cell of animals, we don't have any cell wall. It's only the plasma membrane. And in eukaryotic cell of plants, we also have some thylakoids which are responsible for photosynthesis, and there is a cell wall which is mostly provided from celluloses, which produce some mechanical protection of these cells. But the animal cell, we don't have any cell wall, just keep in mind. Okay. First structure which we are going to talk about is nucleus. The main function of nucleus is to separate and protect DNA from cytoplasm. It's so now we are going to talk about eukaryotic cell only, because these cells are present in humans, and this course is mostly focused on the biological aspects of human cells and biology, which is mostly focused on humans or animals at least. So nucleus, it's a as you can see, it's not just a flat membrane which surrounds the genetic material. However, here, what's happened? Why it is switching? I don't want it. Okay, so here you can see, maybe I can use a pointer. Okay, so here actually you can see on this in this picture you may see two organelles. The first one is nucleus. Maybe I will just circle it with the red color. The membrane protects the genetic material. This is first point. And this part, this blue one with the red dots, it's endoplasmic reticulum, which is in close distance to every nucleus. The reason is proteosynthesis, because in nucleus we have genes, we have our DNA. DNA serves as a recipe, as a book of recipe for all proteins. If we would like, or our cell or our body would like to synthesize a single protein, the signal is coming to the nucleus just to rewrite the information about the content of this protein to messenger RNA. This messenger RNA is rewrite into the, so DNA, the information in DNA is rewritten to mRNA, mRNA is released from nucleus and serves as a recipe for the production of proteins in ribosomes in endoplasmatic reticulum, roof endoplasmatic reticulum. So it's logical that endoplasmatic reticulum, which contains many ribosomes, has to be in close distance to the nucleus. As you saw in the video, cells or the structures of the cell, I mean the molecules, they are able to move. We have plenty of biological rotors and motors and molecules which can be moved one by one, step by step. Recently, this topic is very highly discovered by many scientists around the world. It's a very challenging topic. It's not easy to study these rotors or molecular rotors, or we are talking also about the molecular robotics. Also, scientists are trying to get some inspiration to synthesize these molecular rotors and just to use these inspiration for production of some chemical reactors or to mimic nature. Yeah, to mimic nature, this is the biggest challenge ever. So this envelope serves for the protection of nucleus. About DNA, we are going to talk, maybe, I don't know the number of the lecture, but definitely we are going to talk about the prokaryotic as well as eukaryotic DNA separately. We have separate lecture to talk about the DNA and RNA and also proteosynthesis, so do not worry. This lecture is pretty basic, and the main aim is maybe to repeat all the information which I expect that you have from your high school, and maybe a little bit equalize the knowledge from these basic things. So I expect what I said, I expect that you already know. So endoplasmatic reticulum, we have two, rough endoplasmatic reticulum and smooth endoplasmatic reticulum. I already mentioned that endoplasmatic reticulum closely surrounds the nucleus because of the proteosynthesis. This is a system of vesicles which are interconnected. The reason why these vesicles are applied, it's not a flat, it's really like a labyrinth. And if you just look at this structure in detail, you will see why we need this very complicated and complex structure. Because many biochemical reactions, they are not provided in solvent, in a liquid, actually not in water solvent. That's really a bit overdone expectation. Majority of the biochemical reactions are provided on the surface of some membranes. Usually, that's the reason that mitochondria, endoplasmatic reticulum, and Golgi, they have this vesicle structure. So many times the membrane which creates these vesicles is enveloped, and this strategy increases the inner space of these organelles and allows to separate the biochemical processes and also to enlarge, to extend the space which can be dedicated to provide some biochemical reactions. Endoplasmatic reticulum, we have two. The root one, it's firstly used or primarily used for the biosynthesis of protein. I mean the primal structure of them. So amino acid, so to synthesize the polypeptide chain. The most responsible organelle is a ribosome. You may see ribosomes like these little dots here. We are going to talk about ribosomes also in detail. And also inside endoplasmatic reticulum, after the production of this polypeptide chain, so inside in these vesicles, the polypeptide are modified to become three-dimensional structure, to create some disulfidic bonds, to add also some non-protein part like some metals, sugars, and so on. So it's like a factory which finalize the protein structure. You may imagine this process like that we prefabricate in ribosome some structure of a protein, which is quite simple, like a doll. So we have a doll, but the doll has no eyes, no hair, no lips, nothing, just the prefabricate of a doll, just it has a shape. And inside endoplasmatic reticulum, this doll is receive hair, receive eyes, and also it's colored, maybe dress up also. The smooth endoplasmatic reticulum, it's in close relation with smooth endoplasmatic reticulum, but mostly mostly endoplasmatic reticulum, smooth endoplasmatic reticulum is responsible for the synthesis of lipids. This is the first aim, and also it regulates the content of calcium. So probably you know from physiology and other subjects that calcium is important for the regulation of the plasma membrane potential and also for the regulation of muscle contraction. We are going to talk about it from the biological point of view. So also next time, not next time, but in the future, this semester. And now we are going to talk about ribosomes. Ribosomes, its structures, little structures present, they have two destinations. The first one, they sit on the surface of root endoplasmatic reticulum, or they are flotating in cytoplasm. So these two types of ribosomes, but they have the same structure. The structure is combined from ribosomal RNA, so it's RNA and proteins. It has two subunits, the big one, and the small one. The small between the small subunit and the big subunit, there is a marrow, which serves for the clapping or capturing of messenger RNA. So the recipe, how to build up the protein. So the information written in messenger RNA gave us the information about the sequence of amino acids which is going in the polypeptide chain one by one. So methionine, cysteine, and so on. So this information is based on the triplicates of this triplet combination of bases, which are the main part of the RNA structure. About this process, we are going to talk detail also in the future. So here is the factories for proteins, ribosomes, another cell structure. And now we are getting to Golgi, Golgi complex or Golgi apparatus. This organelle was developed by Camillo Golgi. It was Austrian scientist, so from our country, because Czech Republic was a part of the Austrian Empire together with Hungary, Slovakia, and Austria. And he died, I think, in the first part or first quarter of 20th century. So he lived or were active in the border between the end of 19th and the beginning of 20th century. And he also received the Nobel Prize, and he's pretty famous by discovering of this organelle. So Golgi is also very important organelle in the protein proteosynthesis. The main aim of Golgi is the packaging of proteins. Golgi creates just to put together the same proteins. So for example, insulin just into one packet. It's just some spedition of these proteins. And also the same proteins are put into the packages, which is called vesicles or somes vesicles, and by this way in somes or vesicles they are transported to the target destination. There are, of course, various target destination of proteins. They may remain in the plasma membrane as an integral membrane protein, or they may be transported from the cell to some target destination. Just an example, some hormones or protein hormones which serves as a signalization, or they are part of the signalization system of every cell. So that's it. Also, Golgi is important for the saccharide synthesis. Especially this has a great potential in plant cells. Also, the molecules which are coming to the Golgi, I mean the proteins which are coming there from endoplasmatic reticulum, they became sulfated, and also the three-dimensional structure of proteins, it's polished by these reactions. And that's also the saccharides are added to the structure of proteins to create proteoglycans or glycolipids. Another reaction which occurs there is the phosphorylation, so glycosylation and phosphorylation. Phosphorylation is the addition of phosphate group to the structure of proteins. It's another way. By this way, the proteins which are produced, these proteins mostly serves also for the signalization system of the cell, and the main destination of such the produced proteins is also the cell membrane. Okay, maybe that's the main. Another way, another point of Golgi, it's apoptosis. You probably know that the main role in apoptosis is mitochondria. Also, Golgi is very important because some proteins which are sensitive to some stress for cell, it's localized in Golgi as well as in mitochondria. So Golgi helps mitochondria to provide apoptosis. Apoptosis is cell death. It's very important in the, for example, cancer management, immune system reaction, and so on. Now we are getting to maybe the most important organelle within the cell, mitochondria. Mitochondria has a quite interesting position and role within the cell because it has own DNA. This DNA is circular. It has the same character as a prokaryotic DNA, and therefore it is expected that mitochondria firstly was a prokaryotic cell, separate prokaryotic cell. But in the past, when the eukaryotic cells were developed, one cell just eaten mitochondria as a prokaryotic cell. However, not eaten completely. Mitochondria remain inside eukaryotic cell as a much smaller organism. So therefore, we call mitochondria like a semi-autonomous organelle. It has two cell membranes. The first one is smooth, and the inner one it's a little bit more. It's like root, or it has it increased the inner space of mitochondria. It's created from crystals, so this inner membrane is enveloped. From the biochemical point of view, this mitochondria, they serve like factories for electricity. ATP, all the respiration, all the respiration cell, the respiration chain, as well as the Krebs cycle, both these processes are situated in mitochondria. Without mitochondria, the eukaryotic cell has practically very low access to energy, which can be used for many biochemical processes and reactions. You may imagine mitochondria like Yeah, like a factory for electricity, because it's a really specialized organelle for the energy production. Also, many enzymes which are essential for the energy production are placed, or the genes for these enzymes are written in mitochondrial DNA, sorry, in mitochondrial DNA. That's another point. And also, I have also mentioned one thing I have also in my mind. Mitochondria, if we have a children and if we inherit DNA from nucleus, it's approximately inherited 50 to 50% from father, from mother. So the child which is going to be born from two people, mother and father, it has 50% of DNA from father, 50% from mother. However, this presumption is not valid for mitochondria. Why? If we have eggs and spermias, how it looks like spermia? Okay, so it has just a half of chromosomes, so 23. Eggs 23, spermia 23 is the same. Mitochondria's, spermias are smaller. It contains only the chromosomes, 23 chromosomes, and also flagellum. But for the movement of flagellum, it's very essential to have a very rich part to mitochondria to supply the energy for the movement. And these mitochondria are present in the neck of spermia. And during the fertilization, the process, so we have egg and sperm, and once the sperm is reached egg and captured and trapped by egg plasma membrane, we can observe that that egg plasma membrane is clumping the spermia before egg. So only the hat, which contains the genetical material coming from nucleus, is getting inside the egg cell. All the mitochondria coming from the father, from spermia, are remaining on site. So it means that all the mitochondria which we inherited is only from the mother. And therefore, mitochondrial DNA can be used for the generation of the mother heredity trees. And I think this topic is pretty interesting, all the genetic heredity and so on. Therefore, we have a special lecture about the genetics. So I think that it's the last lecture before Christmas or after Christmas. So that's it. Also, mitochondria plays an important role in aging. All the disorders of mitochondria, about some depletion of oxygen, toxic presence of toxic elements, toxic chemicals inside the cell. Mitochondria are pretty sensitive on these changes, and therefore mitochondria play the crucial role in apoptosis or in regulation of the cell life. Okay, I think this is enough about the cell structure or cell organelles. Now I would like to ask you, here I'm going to open the Excel, and please, you saw just the general characterization of every organelle within the cell. Now, please tell me which organelle suits more to you and you would like to prepare a presentation for next time. So what about Golgi complex? Who would like this topic, Golgi complex?
Can I do?
Just name, please.
Mariam Al-Sibai.
Mariam. Okay, Golgi. What about lysosome? would like to prepare presentation to lysosome. So you may get some extra points. If your presentation will be super, you will receive 3 points. If your presentation will be on average, you will receive 2 points. If your presentation will be so, just 1 point. So everything depends on your effort which you put to the presentation. The presentation, it's not long, 3 minutes. Just prepare 1 slide, put there the picture of the organelle and prepare a few keywords and then your speech. 3 minutes, that's it. Okay, mitochondria. Who would like to prepare presentation to mitochondria?
Can I do it on my
name?
Sanskriti.
Yes. Again.
It's
Name.
Sanskriti.
Sunny.
Yeah, thank you.
Yeah, because maybe the signal is wrong. Okay, Sunny will have mitochondria. What about nucleus?
I'll take the nucleus, if that's okay. Marek?
Okay. What about endoplasmatic smooth endoplasmatic reticulum?
I'll take the endoplasmatic reticulum.
Name? Didn't catch your name. Please, who would like to have endoplasmatic reticulum? I didn't catch the name, sorry.
Eva.
Eva, okay.
Yeah.
So, ribosomes.
Ribosomes.
Katerina?
Yes.
Yes. At the end of the semester, I'm going to recognize you based on your voice even. So, and the last, okay, smooth endoplasmatic reticulum. So we have just these 2 topics, lysosome or endoplasmatic reticulum. Maybe everyone who is here just has a topic, okay.
Yeah, I choose, can I choose lots endoplasmatic?
Okay, yes, sure. Okay. Okay, lysosome remain, and also Omar, maybe he's not here, and doesn't matter. Okay, yes, I have here 6 students. Yeah, 6 students, but yeah, okay, that's fine. Now I'm going to stop the recording because I think it's enough. And now I'm going to show you 15 minutes video, which also compress together all information or just repeat all information about the cell. So now I just repeat the information which I expect that you have from high school. Now you have your own topic. Now I'm going to show you the video and the time which is remaining you may use for the preparation of the presentation. So what I expect from you, just prepare 1 slide, something like that, just 1 slide, just put there a picture. Okay, this is for plasma membrane. No one has this one because I have a little bit longer presentation for plasma membrane, because plasma membrane is very important for another lectures which we have. So just found 1 nice picture of the organelle you have chosen. Then you found the information why this organelle is important for the cell metabolism, for the cell life, for the cell structure, which processes are occur there, what is the risk of this membrane, and so on. Yeah. The presentation should be only 3 minutes long, not longer, just 3 minutes. The information should be the most important one, most crucial one. I hope that you will now just fix the information about the cell structure, because you will put inside some effort and you have to prepare something which is your own. So is it clear? Everybody knows what to do for next time? Just maybe your okay. Yes, raise hand and it's fine. Great. This is also okay. Great. So let's see the video. Yeah, I'm going to.